Hello dear student, welcome back to my channel once again. In my last video, we have studied structure of secondary oocytes. Today we will discuss most important phenomena of this topic is fertilization. Guys, I think all of you know about fertilization because fertilization concept we are already studied in the first topic of our syllabus, but we study in reference to plant. But today we will see fertilization in human. So let's start our today's point. The fertilization involves the fusion of sperm nucleus and the egg nucleus. That is the sperm pronucleus and the egg pronucleus, and restore the diploid number of chromosomes. The fertilization ovum form is called zygote. That is, it maintains the or it restores the diploid number of chromosomes. Means the sperm pronucleus carries 23 number of chromosomes and the egg pronucleus carries the 23 number of chromosomes. Together, the zygote contains the 46 number of chromosomes. In human being, the fertilization is internal. In human being, the fertilization is internal and occurs high up in the fallopian tube. It is occurred in the fallopian tube or it is occurred in the uterine duct. The fertilized egg or also called as the zygote will develop into an embryo. This process occurs within the uterus. As in the fertilization, it is occurred in some mechanism. It occurs in some mechanism. So what is that? The mechanism of fertilization. Semen released during the ejaculation. Ejaculation has sperm and some secretion. The coagulated semen now undergoes liquefaction and the sperm become active. The mechanism of fertilization is as follows. The first point we see, that is the movement of sperm towards the egg. Second one, entry of sperm into the egg. Third one is acrosome reaction. Fourth one is activation of sperm. And the last one we see, that is the fusion of sperm and the egg. The first point we see, that's the movement of sperm towards the egg. As in the movement of sperm, it involves capacitation of sperm reaching the vagina. Capacitation का मतलब क्या है? As a sperm undergoes a number of changes, that is called as the capacitation. It called as the capacitation. This image indicates a phenomenon of capacitation. As when the sperm it is present in the epididymal region, there is a presence of the some structure around to the head region of the sperm. But when the sperm it is ejaculated and it is ejaculated, that time this structure it become thick. You can see the outer side structure. This outer side structure it is present around to the sperm head. That is when it is ejaculated. But when the sperm it is enters in the female genital tract, that time this outer structure it become dissolved or it is removed from this head region of the sperm. That phenomena it is called as the capacitation. That is, the sperm undergoes a number of changes. The sperm undergoes a number of changes. As when the sperm it undergoes a number of changes, means a capacitation it's occurred. Here, as many as 50% of the sperms is demotilized. It is broken or destroyed, and remaining sperm undergoes capacitation phenomena, that is the changing the structure of the sperm. For the capacitation, it requires near about 5 to the 6 hours. Across the membrane, it becomes thin. Calcium enters the sperm and the tail begins to show द रैपिड whiplash मूवमेंट. यहाँ पे जब ये कैपेसिटेशन की फिनोमिना कंप्लीट होते हैं तो ये स्पर्म के अंदर रैपिडली ये जो स्पर्म है ये फैलोपियन ट्यूब के अंदर, यूटेराइन ट्यूब के अंदर और ओविडक्ट ट्यूब के अंदर चले जाते हैं. एज अ रिजल्ट ऑफ कैपेसिटेशन द स्पर्म become extra active and to start moving towards the vagina, towards the uterus, or it is reaches up to oviduct. The prostaglandins which is present, this prostaglandin activate the sperms. The vestibular secretion of the sperm, which is the vestibular secretion of the female, also enhance the sperm motility. The sperm swim at an average speed of 1.5 to 3 mm per minute. Sperm reaches up to the ampulla region. This region it says as the ampulla region. As a result of the own swimming and partially by the contraction of uterus, the fallopian tube stimulate the oxytocin of the female. After capacitation, the sperm may reaches, ये जो sperm है ये ampulla region तक आ जाता है within near about the 5 minutes. And this sperm it is remain viable near about 24 to 48 hours. As the when this first phenomena is completed, what's that point we discussed? It involves the capacitation of the sperm reaching the vagina. Here, as many as 50% are demotilized, broken, destroyed, and remaining sperm undergoes capacitation. Remaining sperm undergoes the capacitation. This process requires near about the 6 to this process it requires near about the 5 to 6 hours. It requires 5 to 6 hours. Acrosomes membrane become thin. Calcium enters the sperm and the tail begins to show the rapid whiplash movement. As a result of capacitation, the sperm become more active and begins to start moving upward from vagina towards uterus and reaches up to the oviduct. The prostaglandin it is also activate the sperms. The vestibular secretion of the female also enhance the sperm motility. The sperm reaches up to ampulla as a result of the own swimming and partially by the contraction of uterus and the fallopian tube. And this sperm it is viable near about 24 to 48 hours. Afterward that the second mechanism we see, that is the movement of sperm towards the egg. This is the first point we see. The second one we see, that is the entry of sperm in the vagina. Entry of sperm into the egg. What's that point is it? Out of the 200 to 400 million sperm, only few hundred manage to reach the ampulla region. बहुत ज्यादा नंबर के स्पर्म होते हैं 200 to 400 millions of sperm, लेकिन उनमें से सिर्फ few hundred of sperms it is reaches up to the ampulla region. So many sperm reaches the ampulla, but only a single sperm it is fertilized. It's a fertilize the ovum. सिर्फ सिंगल स्पर्म ये ओवम को फर्टिलाइज करेगा. The sperm after reaching the egg or ovum come to lie against it. The sperm acrosome, ये जो sperm के ऊपर का जो भाग होता है acrosome, this acrosome, this says as the acrosome, ये acrosome होता है. When the sperm it is reaching the egg or ovum come lie against it, the acrosome released, ये acrosome क्या release करता है, acrosome released lysine, hallouridinase and the corona penetrating enzyme. They separate and dissolve the cell of corona radiata. So the sperm head passes through the zona pellucida. ये जो sperm का head होता है, acrosome का region होता है, उसमें लाइसिन, हेलोयूरिडिनेज और कोरोना पेनिट्रेटिंग एंजाइम्स ये रिलीज होते हैं. रिलीज होने के बाद क्या होता है, वो जो कोरोना रेडिएटर का लेयर होता है ये डिसॉल्व हो जाता है. दिस वॉल इट बिकम डिसॉल्व. सो द स्पर्म हेड पासेस, जो स्पर्म का हेड होता है ये जोना पेल्यूसाइडा के पास आके रुक जाता है. एज द स्पर्म and they separate and dissolve the cell of corona radiata. So the sperm head passes through zona pellucida of the egg. The zona pellucida has the fertilizing receptor protein. ये जो zona pellucida होता है उसके अंदर होता है fertilizing receptor protein. लेकिन ये fertilizing क्या है, anti fertilizing अभी हम आगे पढ़ेंगे ये क्या है. As the F.R. Lilly proposed the fertilization theory in 1914, which is based on the sea urchin, that is an echinodermata. This point we are already studied in the 11th standard. According to this theory, the egg ovum secrete the chemical name as the fertilizing. ये जो egg होता है यहाँ पे कौन सा chemical secrete होता है, वो होता है fertilizing. And this fertilizing it is composed of the glycogen, and this glycoprotein consists of monosaccharide and amino acid. And the sperm head, it is the sperm head surface contains the protein substance that is called as anti-fertilizing, and this anti-fertilizing it is consist of the amino acid. जैसे कि हमने देखा कि एंजाइम्स में हमने देखा कि ये लॉक एंड की कंसेप्ट होता है, जो स्पेसिफिक प्रोटीन होता है वो स्पेसिफिक एंजाइम्स लगता है उसके लिए. वैसे ही यहाँ पे जो एंटी फर्टिलाइजिंग होता है उसको स्पेसिफिक फर्टिलाइजिंग लगता है. ये जो कंसेप्ट है ये प्रपोज की है F.R. लिली ने. एज व्हाट्स दैट fertilizing and anti-fertilizing. The zona pellucida, ये जो zona pellucida होता है उसके अंदर होता है fertilizing, and this fertilizing receptors the protein. There is a presence of the fertilizing receptor protein. The fertilizing it is bind to the specific protein. इसके लिए fertilizing किसको bind करेगा, वो specific protein को ही bind करेगा. वो specific protein किसके अंदर होता है, वो होता है स्पर्म के हेड के अंदर. दैट इज कॉल्ड एज द एंटी फर्टिलाइजिंग. इट ब्रिंग्स अबाउट द कॉन्ट्रैक्शन ऑफ द स्पर्म. ये फर्टिलाइजिंग और जो एंटी फर्टिलाइजिंग की रिएक्शन होती है ये बहुत ज्यादा स्पेसिफिक होती है. That is, thus the fertilizing anti-fertilizing reaction it is called as the compatible reactions. It called as the compatible reaction. As when the sperm it is head in when the sperm head it is come in contact with the zona pellucida, there is a release of the sperm lysine. It is released a number of chemicals. That is, first one it's a hallouridinase, it's released. Then the corona penetrating enzyme is released, zona lysine is released. And when this chemicals it is released, the acrosome मेम्ब्रेन आप देख रहे होगे यहाँ पे जो एक्रोसोम का स्ट्रक्चर है ये क्या हो गया, यहाँ पे द एक्रोसोम मेम्ब्रेन टर्निंग इनसाइड आउट. दिस एक्रोसोम मेम्ब्रेन इट इज टर्निंग, ये जो एक्रोसोम का मेम्ब्रेन होता है ये टर्निंग इनसाइड देन आउटसाइड. This is a significant phenomena. It is occurred when this sperm lysine it's released from the acrosome. ये यहाँ पे सबसे ज़्यादा important point है. Afterward, what happened? The acrosome reaction. As the sperm head touches the zona pellucida, ये जो sperm का head होता है, ये zona pellucida को touch करता है. As the sperm head when touches to the zona pellucida in the animal pole region, its acrosome covering it become rupture. ये जो covering है ये rupture हो गया. acrosome covering it's a rupture. Its acrosome covering its rupture to release the enzyme that is the acrosin, or this acrosin it is also called as a lysine. यहाँ पे ये lysine released हो गया, acrosin released हो गया. They act on the zona pellucida. ये कहाँ पे act होगा? ये जो येलो कलर का दिख रहा है आपको पोर्शन, ये है zona pellucida का लेयर. और this lysine or the acrosin it is act on the zona pellucida at the point of contact, thus cause the egg reaction. A small fertilization cone is formed on the egg membrane. The sperm head in contact with that cone. ये जो sperm का head है ये इस zona pellucida के layer को contact हो गया है. As the sperm head come in contact with this cone, it come in contact with the cone. This result in the production of the weak way of depolarization. The plasma membrane of both the cell, that is the egg plasma membrane और sperm head plasma membrane, it is dissolved. ये जो egg का plasma membrane होता है और sperm का plasma membrane होता है ये dissolve हो जाता है जहाँ पे ये contact हुआ है. जहाँ पे ये contact हुआ है वहाँ पे क्या होता है? ये membrane है dissolve हो जाता है. It is dissolved at the point of contact. At the point of contact, the sperm nucleus that is says as the nucleus the sperm nucleus and the centriole. यहाँ पे दो centrioles होते हैं. पहला होता है proximal centriole और दूसरा होता है जो वो होता है distal centriole. The sperm nucleus and the centriole, ये जो smaller portion है वो है centriole का portion. It enters the egg. ये दोनों भी जो पोर्शंस होते हैं स्पर्म के हेड के अंदर होते हैं ये दोनों भी अंदर आ जाते हैं. वाई द अदर पार्ट जो है मिडल पीस है टेल पीस है इट्स रिमेन आउटसाइड. एज सून एज द स्पर्म हेड टचेस द वाइटेलियन मेम्ब्रेन, ये जो स्पर्म वाइटेलियन मेम्ब्रेन को टच करेगा the cortical reaction get activated and changing the vitelline membrane it into the fertilization membrane by deactivating the sperm receptor of the zona pellucida. The distinct perivitalian space is created around the fertilization membrane. So, it prevents the entry of the other sperm. इसकी वजह से ये जो बाकी के sperm इसके आजू बाजू होते हैं वो अंदर नहीं आ सकते. What about the further part is here? अभी तक हमने क्या देखा है? एक्रोसोमल रिएक्शन देखा है. एक्रोसोम में क्या देखा हमने? As the sperm head touches the zona pellucida in the animal pole reaction, its acrosome covering is ruptured to release the lytic enzyme. That lytic enzyme it called as the acrosin or the lysine. They act on zona pellucida at the point of contact. They cause the egg reaction. A small fertilization cone is formed. The sperm head touch in contact with this cone. It results in the production of the weak way of depolarization. The plasma membrane of both the cell, that is the sperm head and the egg plasma membrane, it is dissolved at the point of contact. The sperm nucleus and the centriole it is entered inside the egg while the other part it remain outside. As soon as the sperm head touches the vitellian membrane into the fertilization membrane by deactivating the sperm, that is deactivating the sperm receptor of the zona pellucida. A distinct perivitalian space is created around the fertilization membrane. This prevents any further entry of the sperm into the egg. That is, it avoids the polyembryony. It avoids the polyembryony. The further part we see, that is a fourth one, it's a activation of the sperm. The sperm, sorry, the ovum become fertilization as and was at metaphase second. The ovum before fertilization, ये ओहम फर्टिलाइजेशन होने के पहले ये कौन से फेज में था? ये था मेटाफेज सेकेंड फेज में. ये हमने लास्ट वीडियो में भी देखा होगा. As it is present in the metaphase second with मेटाफेज सेकेंड स्टेज विथ अ कॉन्टैक्ट ऑफ स्पर्म हेड. जब ये स्पर्म का हेड वाइटेलियन मेम्ब्रेन को टच करता है, that time what happen? The ovum, it become activated, and it is due to the activity it resumes and it's complete it meiosis second division. When the meiosis second division, whenever it is there, with what happened, there is a formation of the second polar body and a single haploid oval structure is formed, that is called as the female pronucleus, and this female pronucleus, it says as the true ovum, or it called as the egg. Afterward, after the formation of the female pronucleus, what happened? There is the fusion of the egg and the sperm. The covering of male and the female pronucleus degenerated. ये जो male pronucleus और female pronucleus के ऊपर जो कवरिंग होती है वो क्या होती है? वो degenerate होती है. And what happened? The chromosomal pairing it start. When the chromosomal pairing it start, that is called as the synkaryon. Syn का मतलब क्या है? Fusion. Karyon means nucleus. That is the chromosomal pairing it start. Karyogamy it start. Karyogamy. Karyo it stand as a nucleus and gamete it stand as a fusion. That is the mixing or the chromosomal pairing it start, and result is what? Result is the zygote is formed. Zygote is formed from the chromosomal pairing of the female pronucleus and the male pronucleus. What is the role of the proximal centriole present in the nucleus? It is remain as it is. The proximal centriole, it receive from the sperm. It receive from the sperm help in the formation of a synkaryon spindle and cleavage. Cleavage means what? Division. Breaking. Division of cell into the two blastomere. That is this proximal centriole. It plays a very important role in the cleavage of a cell. So what is the significance of this fertilization? So all these phenomena we have studied. The first one, what is the first image indicate? It indicates after the sperm penetrate the secondary oocyte. The oocyte completes the meiosis second division, forming the ovum and the second polar body is formed. The sperm and the ovum nuclei, it is swim. The sperm and the ovum nuclei, it is swell, forming the pronucleus. The pronucleus approach each other and the mitotic spindle forms between them. There is a formation of this spindle fiber. This spindle, it is formed due to the presence of the proximal centriole. That is, the chromosome of the pronucleus intermix. जो chromosomes होते हैं वो intermix होते हैं and the fertilization is finished. That is, there is a formation of the zygote. As after the formation of zygote, the proximal centriole, it is start the first cleavage, and there is a formation of the two cell structure that is called as the two cell stage blastomere. So what is the significance of these? The first one is the secondary oocyte complete the process of oogenesis and transferred it into the mature ovum. The diploid chromosome number it is restored. ये जो diploid number के chromosomes है वो restored होते हैं in the zygote by the process it called as the syngamy, that is a fusion of the nucleus. The ovum lacks the centriole. ovum के अंदर centrioles नहीं होते. It's a necessary for the further division are received, and this centriole it is received from the sperm. ये जो centrioles यहाँ पे लगते हैं वो कहाँ से आते हैं? It is received from the sperm, that is the proximal centriole. This proximal centriole it plays a very important role in the fertilization phenomena. The fertilization involves the fusion of male and the female gametes and forms from the two parent. It result in the variation which is significant to evolution. यहाँ पे क्या होने वाला है? यहाँ पे दो पेरेंट्स इन्वॉल्व होने वाले हैं. दो पेरेंट्स में से जो स्पर्म और एग ऑब्टेन होने वाला है उससे क्या होने वाला है? उससे फर्टिलाइजेशन होने वाला है. फर्टिलाइजेशन होने के बाद क्या होता है? जो न्यू इंडिविजुअल फॉर्म होने वाला है वो इन दोनों से अलग रहता है. वेरिएशन इज देयर, एंड व्हाट इज दैट? ड्यू टू द वेरिएशन there is the evolution is there. So what about the concept map? As there is some physico-chemical events it is occurred in the fertilization. The first one is as the sperm reaches the ovum, it bring about the sequence of events. First one is breaking of membrane covering the acrosomal region. Next one is release of chemical substances or enzymes called as the sperm lysine from the acrosome. This sperm lysine हमने अभी देखा कि sperm lysine में कौन कौन से chemical थे. As there is a presence of the hallouridinase, there is a release of corona penetrating enzyme and there is a release of the zona lysine. As this hallouridinase, what is the role about the hallouridinase? This hallouridinase an enzyme that act and dissolve the ground substances bind the follicle cell of ovum together and forms corona radiata. From corona radiata, the corona penetrating enzyme, ये जो corona penetrating enzyme इसका रोल क्या है? That dissolves the corona radiata region. ये जो corona radiata की सेल जो होती है उसको डिसॉल्व करने का काम करता है कौन सा पोर्शन? इसको करता है डिसॉल्व कोरोना पेनिट्रेटिंग एंजाइम. The third one, its chemical that is known as the zona lysine. Zona lysine, or it is also said as the acrosine, and enzyme that helps to digest the zona pellucida region. Zona pellucida region को जबकि स्पर्म का एक्रोसोम ये जोना पेल्यूसाइडा के रीजन तक जाता है तो वहां पे जोना लाइसिन रिलीज करता है. जोना लाइसिन रिलीज होने के बाद वहां पे क्या होता है? जोना पेल्यूसाइडा डाइजेस्ट हो जाता है. द fertilin protein present on sperm surface act as a egg recognition protein to recognize the gamete of the same species. So here we say that is the fertilizing anti-fertilizing is the species specific which is put forth by the FR Lilly. As the acrosome come in contact with the zona pellucida, जब ये एक्रोसोम जोना पेल्यूसाइडा को कांटेक्ट में जाता है तब क्या होता है? द एक्रोसोम टर्न इनसाइड आउटवर्ड. जब हमने देखा कि यहाँ पे जो एक्रोसोम होता है ये ऐसे inside to outside turn होके बाहर आता है. It is turn inside and the outside and release the chemicals that dissolves the vitalian membrane and the plasma membrane of the egg at the point of contact. The entry of the sperm in the egg cytoplasm and the entry of more sperm, it is prevented by the chemical changes on the surface of the egg. Okay, I think so you understand this fertilization phenomena. In the next video, we will see about the embryonic development. Thank you for watching my video. Thank you.